Hello, dear students. In this video, we're going to continue our chapter about the nervous system, and we will start about the nervous system, part 2. Let's have a quick revision about the notes that we have started before. You have to know that group of nerve fibers, they represent a neuron. Group of neurons, they give a nerve, and group of nerves, they compose what's known by the nervous system. Also, let's have revision about the different types of neurons. They are classified according to their function, according to their structure. According to their structure, you have a unipolar neuron, T-shaped neuron. We have the bipolar neuron, and we have the multipolar neuron having many dendrites and only one axon. According to their function, we have the sensory neuron, it's called the afferent. It receives the information and receives the message and conduct the information, which is sensory one, from the peripheral nervous or peripheral sensory organ to the central nervous system toward the brain or the spinal cord. Associative neuron, anterior neuron, it receives nervous message from the sensory neuron, it integrated, analyze it, and then it carries the message toward the motor neuron. So this one, it's like a link. Mainly it's found inside the brain and the spinal cord. Number 3, the third type of neuron according to their function, it's the motor neuron. It's called the efferent. It carries the message away from the central nervous system toward the effector organ, whether a muscle or a gland, and this will induce a response. So here, as you see, the efferent neuron, it carries the message from the organ, from the sensory organ toward the central nervous system, the brain and the spinal cord, and the efferent, it carries it from the central nervous system toward the effector organ, whether a muscle or a gland. This is called efferent or motor neuron. In between, we have the interneuron, as I said, it's called associative neuron. It's like a link between the sensory and the motor. Between the link is found in the brain and the spinal cord only. Actions, they are two types. We have actions that require thinking and actions that does not require thinking. Actions that require thinking, they require the intervention of the brain and the spinal cord, whereas action that doesn't require thinking, the action that doesn't require thinking, it also doesn't require the intervention of the brain. Action that require thinking, that we need to think before we act them, is called voluntary actions, whereas those that do not need the thinking or the intervention of the brain, they are called involuntary actions, and they are called reflexes from now on. Reflexes, this is the word here. Factors that affect the speed of propagation of a nervous message in a nerve fiber. We have the diameter. As the diameter increases, the speed of propagation of the nervous message increases. This is the first factor. We have the myelination. So the presence of myelin has higher effect on increasing the speed of the propagation of the nervous message than that of the diameter, because it will move, the message will move in a saltatory manner, with jumping like this, and the nodes of Ranvier, if you remember, the gaps here between the myelin is called nodes of propagation of the message. As I said, it moves in a saltatory manner, in a jumping manner. It's called saltatory. So myelin is an insulator, so it doesn't conduct a nervous message. So the message will jump the myelin, like shown in this diagram. So here, the propagation of the nervous message will be in a saltatory manner, a jumping manner. So let's start with some experiments. Aim of the following experiments is to study the state of a neuron at rest, and to be more specific, the electrical state. Here we have in experiment 1, we have the voltmeter. This voltmeter is connected to the external sides, as you see here, external sides of the axon of the nerve fiber. The needle of the voltmeter did not deviate from zero, so it shows 0 millivolt delta V. So what does this imply? This means that there is no potential difference delta V. There is no potential difference across the surface of the axon, or there is no potential at all at the surface of the axon. So at the surface, both needles, they are placed at the surface of the axon, and this axon is placed in a physiological solution. So both of them, they are placed on the surface as seen in this figure. So both of them, they are placed on the surface. Potential difference delta V, it's equal to 0 millivolt. What does this indicate? This indicates that there is no potential difference across the surface of the axon, or you can say there is no potential at all at the surface of the axon. Experiment 2. The needle of the voltmeter did not deviate from zero also in the second experiment. Second experiment was the difference between the first and the second. The first experiment, both needles, they are placed on the surface of the axon, here on the surface of the axon. But here in experiment 2, both of them, they are placed across the surface of the axon. Here there is no potential difference at all. Moving for experiment 3. In experiment 3, what do we have? Also, we have an axon, nerve fiber, and we have the needles of the voltmeter. One of them, here the difference, one of them is placed on the surface of the axon, and the second in the internal or the inner side of the axon. So here the potential difference deviates to decrease to minus 70 millivolt as shown here. So the needle of the voltmeter, as seen here, the needle deviates away from zero, and it shows a drop in the delta V in the potential difference. Delta V is the potential difference to minus 70 millivolts. What can we deduce? We can deduce from this experiment, first one, a neuron at rest, meaning that without any stimulation, there exists a potential difference, which is delta V, between the interior and the exterior of the nerve fiber. Pay attention, these at rest without any stimulation, without any excitation. So there exists a potential difference, delta V, between the interior and the exterior of the nerve fiber. Since when we place the needles of the voltmeter, one on the internal side and the second on the external side, or in the interior and the exterior of the nerve fiber, delta V deviates from 0 toward minus 70 millivolt. So the nerve fiber at rest is said to be polarized, meaning that it's negatively charged. Since delta V is less than 0, then the interior of the nerve fiber is negatively charged with respect to the exterior. This is the first two conclusions. So these two conclusions, you have to know them, that the neuron at rest, it has a potential difference that's not equal to 0, it's equal to minus 70 millivolt. At rest, the nerve fiber is said to be polarized, meaning that it's negatively charged. Since delta V is less than 0, so the interior of the nerve fiber is negatively charged with respect to the exterior. You have to know as a note here that the resting potential is the potential difference between the interior and the exterior of the nerve fiber where at rest. That's why it's called the resting potential. Okay, so I repeat, resting potential is the potential difference existing between the intracellular and the extracellular media of the nerve fiber at rest. That's why it's called potential. Okay, so the resting potential, it's polarized, negatively charged, minus 70 millivolt, needles internal side, external, and that's why it Resting potential. There is an electric charge across the membrane of the nerve fiber. This is called membrane potential. This is found across the membrane. So the resting potential, when the cell is not firing, meaning that it's not stimulated, it's at rest, is a 70 millivolt difference between the internal and the external. So the resting potential, from now on, the resting potential of a neuron is equal to minus 70. So this document is showing the resting potential. A cell is not excitable, they are resting. They have the resting potential and charge across the plasma membrane between the internal and the external. The value of the resting potential varies, but it's about minus 70 millivolts. So here, resting potential, as you note, may range between minus 5 to minus 90, depending on the species and the nature of the nerve. Let's move now to the ions and the resting potential. Why do we have resting potential is equal to minus 70 millivolt? Why? It's negatively charged. Ions, they are electrically charged molecules. For example, sodium is a cation, an A+, charge positive. Potassium, K+. Chloride ion, Cl-. The resting potential exists because ions are more concentrated on different sides of the membrane. Since we have Na+ and Cl-, they are outside the cell, while K+ and other organic anions, they are found inside. So the concentration of Na+ outside the cell, this is the cytosol, meaning that inside the cell, in the plasma membrane, and the extracellular fluid outside. The concentration of Na+ outside the cell is 10 times greater than that inside the cell. If you see here, concentration of Na+, it's 150 millimolar, while that in the cytosol it's 15. So in the extracellular it's times 10. Concentration of K+, it's 150, while in the extracellular it's 5, about 30 times. Concentration of Cl- in the extracellular it's 120, while in the cytosol it's 10, so it's times 12. Okay, so what is the origin of the resting potential? Why do we have resting potential? Why it's equal to minus 70 millivolt? First of all, differences in the ionic distribution between the intracellular and the extracellular. We have difference, as we see before in the previous slide, that we have difference in the concentration of Na+, of K+, and Cl- between the intracellular and the extracellular. Number two, we have differences in the membrane permeability to different ions. So it's more permeable, allows the entrance of potassium ions more than that of the sodium ions. This will lead to the electric current across the membrane, which will be the origin of the potential difference. So here, 3 Na+, they are picked from the cytosol toward the cell, and these 3 Na+ will move outside to the extracellular fluid, and 2K+. So here we have 3+, here we have 2+. So we lose 1+ here. 2K+ are pumped in the cytosol, while others, they are moved toward the extracellular fluid. So here, membrane is more permeable. It's more permeable, meaning that it allows the passage of K+ more than Na+. Thus, the inside of the cell will be more negative. So the inside of the cell will become more negative due to the difference in the permeability for the different ions obtained. That's why we have the potential difference, which is equal to minus 70 millivolt. How it's maintained? Due to differences in the membrane permeability to sodium and potassium. Selective permeability, it allows the entrance of K+ more than that of Na+. So it's selective. It selects more K+ than that of Na+. Due to the differences in the ionic distribution between the intracellular and the extracellular, and also due to the action of the sodium potassium pump. It's called Na plus K plus pump. Okay, this is like a pump which allows the fusion of potassium and sodium ions. So to keep in mind, we have some notes, you have to know them. First of all, what the meaning of an excitable neuron? From its name, excitable, a neuron that is able to respond to an effective stimulus. It's called excitable neuron. So a neuron that's able to respond to stimulation. This stimulation should be effective one. So whenever it's excited, it can do a reaction. So it's called excitable neuron. Action potential. Action potential is the term given to the electrical visualization of the response of the neuron. For example, let's give you an example about the action potential, and this will be explained more in details in the coming videos. So action potential, for example, in the cardiogram, electrocardiogram, you see, for example, the heart beats going to be like this. Okay, so these are the action potential. Whenever it's be like this, there is no action potential, meaning that you are dead. Also, some notes about the threshold of excitation. Threshold of excitation is the minimum intensity that is able to stimulate a receptor or may not emit a nervous message or a nervous impulse. Below this intensity, there is no response. For example, I can't feel hot unless the temperature is, let's say, 30 degrees. Below 30 degrees, I can't feel hot. It's normal with respect to, according to me, it's normal. Below 30. Above 30, I will feel very hot. So 30 will be the threshold of excitation. Below 30, I didn't feel anything. It's normal. Above 30, I start to feel hot. So this is an example, a simplification for the threshold of excitation. For example, if I want to do a, let's say, a reaction, okay? A reaction, I need 10 grams of, let's say, sodium bicarbonate. Below 10 grams, there is no reaction. Above 10 grams, the reaction is going to be normal and it works. So here, threshold, it's the minimum level or the minimum intensity needed to cause a reaction. So let's say below 10 ampere or below 10 ampere, let's say below this intensity, I equal 10 ampere, there is no reaction. So 10 ampere is the threshold of excitation. Moving now to upon excitation, we have a change in the potential difference between the interior and the exterior of the nerve fiber. And this should reach the threshold of depolarization in order to initiate an action potential. Threshold of depolarization. What the meaning of threshold? These you have to study them. So at the minimum change in the potential difference that should be reached in the nerve fiber to create an action potential. This is the threshold. I repeat, threshold of depolarization. Depolarization is the minimum change in the potential difference that should be reached. Potential difference delta V to create an action potential. So depolarization, polarization, negativity. Depolarization, moving away from the negativity. So moving toward the positive. Also, to keep in mind, you have to know the minimum time of excitation. Minimum time is the minimum time during which an effective stimulus should be applied in order to excite a neuron. So at rest, the resting potential, as we said before, is equal to minus 20 millivolt. Upon application of intensity I1, I1 is less than the threshold, it's less than the minimum value to cause an action potential. So here there is no change in the potential difference as nothing occurred. So this stimulus is said to be ineffective one. Why? Because it doesn't cause the appearance of the action potential. So in the second experiment, we have application of stimulus of intensity I2. I2, here it's greater than I1, as you see here, but it's less than the threshold of excitation. So here I2 is greater than I1, but it's less than the threshold of excitation. Here we have a decrease in the negativity. We're moving toward the positive. So it becomes greater than minus 70. This is called artifact, but it's still less than the threshold of depolarization. So slight change in the potential difference called slight depolarization. Depolarization means that decrease in the negativity is called hypopolarization. Hypo means that decrease. Polarization, negativity. So decrease in the negativity. We call it artifact. But you have to know both terms. You have to know that it's called hypopolarization, and also you have to know that it's called artifact. Let's make it simple. Hyperactive. Whenever I say, for example, this student is hyperactive. What the meaning of this? Hyperactive meaning that it's very active. It's active higher than the normal. So hyperpolarization, polarization negative. So hyperpolarization, it's increase in the negativity. Hypo is the opposite, is the decrease in the negativity. So decrease in the negativity meaning that we're moving toward the positive. But here we didn't reach the threshold of depolarization. So this will go back to the normal level normally and very fast. So this stimulus is said to be ineffective because it doesn't cause the appearance of the action potential. So here there is no AP action potential. Okay. Moving to the experiment number 3. In experiment 3, we are talking about the intensity I3. I3 here is greater than I2, so sure it's going to be greater than I1. So I3 greater than I2, greater than I1. But the new here, the intensity I3, it's greater than or equal the threshold of excitation. So after applying I3, the intensity I3, we have a decrease in the negativity. So we're going to move to the positive. So we moved from minus 70 to plus 50. So here, minus 70 to the 0, if you want to calculate it as math. So minus 70 to 0, we move to 70 millivolt. Then from 0 to 50, it's 50 millivolt. So 50 plus 70 is equal to 120 millivolt. So here, this is called the action potential. So this is the action potential AP. So we have an action potential. And this can be explained that intensity I3 is called effective stimulation. Okay, also it has another name. We're going to discover this later. So this is the figure. We have stimulating electrodes, S electrodes, positive and negative. We have recording electrode that's connected to the oscilloscope, and we have the reference electrode, B, and this is the recording on the oscilloscope that shows an action potential AP. So upon application of the stimulus intensity I3 greater than or equal the threshold of excitation, there is a change in the potential difference that reaches the threshold of depolarization. So a response or an action potential of amplitude 120 millivolt was recorded. Why 120? It moved from minus 70 to 50 millivolt. So minus 70 to 0, it's 70. Plus 50 millivolt is equal to 120 millivolt. So the stimulus is said to be effective one. Effective, why? Because it causes the appearance of the action potential and is said to be the threshold of excitation. So from now on, the first stimulus that causes the appearance of an action potential is said to be the threshold of excitation. Upon application of stimulus I4, I4 is greater than I3. Also, it's greater than or equal the threshold of depolarization. And we have an action potential. This action potential has the same amplitude. So here, a change in the potential difference occurred that reaches the threshold of depolarization. The same response was recorded. So the stimulus intensity I4 is said to be effective. Okay, so this graph, action potential in a nerve fiber. We have different phases. Number 1 is at minus 70 millivolt. It's called the resting potential. Number 2, this is moving away from the negativity toward the positive. You're decreasing in the negativity, meaning that hypopolarization or artifact. Number 3, we're moving toward the positive, decreasing in the negativity. It's called depolarization. Number 4, we're going to move from the positive toward the negative. So it's called inversion of polarity. Number 5, we have repolarization. We're going back toward the negative. Number 6, we moved to the resting potential, and even less than, we moved to the minus, let's say minus 80 here. It says hyperpolarization. And number 7, here we're going back to the normal state. Going back to the normal state. So number 1, the phases of the action potential, resting potential. Number 2, hypopolarization is called the artifact. Number 3, depolarization. Number 4, inversion of polarity. Number 5, repolarization. Number 6, hyperpolarization. And number 7, returning to the resting potential. This figure describes all the phases of the action potential inside the nerve fiber. So we have 7 different phases. Resting potential. Number 2, hyperpolarization when it increases toward the positive. Depolarization when it moves toward the action potential of 120 millivolt. Inversion, this is at the top, we move from the positive toward the negative. Repolarization, going back to the negative. Returning back to the polarity, returning to the polarization, to the negativity. Hyperpolarization, and finally returning to the resting potential. Okay, thank you. Bye bye.